Everywhere there is life, there are cells. They're too small to see, but they're the basic building blocks of life. Cells come in a tremendous variety of shapes, but most of them share the same basic structure. Cells can live by themselves or with other cells. When they live together, they form cell tissue. Within one type of tissue, all the cells are the same, but living things are made up of different kinds of tissue, each carrying out a specialist role. For example, muscle tissue, nerve tissue, and skin tissue. Most of the organisms we see around us, including ourselves, are multicellular. Cells are mostly very small. You can get some idea of their structure with a light microscope, such as the ones you use at school. To see detailed structures, scientists use electron microscopes. These can magnify the image of the structure many thousands of times. To find out the chemistry of the cell, biochemists use chromatography and many other methods. They've found that there's an incredible complexity of chemicals within the cell. Combining all this information, they've been able to build up a picture of what the cell looks like inside and how it works. They've found out that a cell is like a miniature chemical factory with lots of chemical processes going on at the same time. Different parts of the cell are like different parts of the factory. Cells are all different shapes. They can change shape, and often everything inside them moves around. This is our model cell. The most obvious thing about cells is that they have a boundary around them called the cell membrane. Everything inside the cell membrane except for the nucleus is known as the cytoplasm. The cytoplasm consists of two elements, a semi-transparent thick liquid, the cytosol, and floating in the cytosol, various organelles. The organelles have a wide variety of shapes. Most prominent is the nucleus. This is the control center of the cell. Attached to the nucleus is a system of folded membranes called the endoplasmic reticulum. There are two types. The rough endoplasmic reticulum helps make proteins. The smooth endoplasmic reticulum makes fats. The Golgi complex modifies proteins and produces other chemicals. It then dispatches them to other parts of the cell. The mitochondria convert glucose into an energy-carrying chemical for use all over the cell. Ribosomes make proteins. Lysosomes break down complex chemicals. The cytoskeleton helps support the cell. Now we're going to look in more detail at these different parts. The cell membrane is a masterpiece of design. It's made of a double layer of fatty or phospholipid molecules. It's waterproof, but it has embedded in it protein structures which let selected substances pass through. In this electron micrograph of a white blood cell, you can see other structures projecting from the membrane of the cell. They help the cell to sense conditions outside, such as which cells are nearby. The cytosol is salty water containing large quantities of protein, fat, and carbohydrate molecules. It's fluid, so the chemicals can travel to the various organelles. They can mix together, and they can react with each other. So, the cytosol works as a transport system around the cell. Now let's look at the most prominent organelle, the nucleus. It has a double membrane with pores in it containing special protein. This selectively allows some large molecules to move through. Smaller molecules can pass through the pores by themselves. This is an electron micrograph cross-section of a nucleus. It contains a very special material, DNA, in long stringy fibers called chromatin. The DNA carries all the genetic information about building and running the cell. When a cell divides, the DNA chromatin sorts itself into two sets of chromosomes, one for each cell. So, when it's not reproducing, what does DNA do? Each molecule takes the form of a double helix. The way it is arranged carries information. The DNA is a recipe book for the cell. Let's watch it start some protein manufacture. The instructions are held in the code on a particular part of the DNA. The DNA uncoils at this exact spot. Only one strand is used. Also in the nucleus is the nucleolus, containing another special substance, RNA. Small pieces of RNA form a chain alongside the selected patch of DNA and pick up its code. This chain is called messenger RNA. The messenger RNA then moves out through the nuclear membrane into the cytosol, where the chemicals and organelles await instructions from the center. The DNA strands left behind can now coil up until the next time they're needed to make messenger RNA. The ribosomes float around in two parts. The two parts clamp themselves around the messenger RNA to form a complete ribosome. As the ribosome moves along the messenger RNA, it selects different amino acids from the cytosol. The message on the RNA tells the ribosome which amino acids to select as building blocks to make the protein. And so, as the ribosome moves along, a strand of protein is made. This is released out into the cytosol. Some ribosomes don't float around. They're attached to the rough endoplasmic reticulum. The reason it is rough is the ribosomes form pimples all over the outside surface. This is what the rough endoplasmic reticulum looks like under the electron microscope. As before, the RNA tells the ribosome which amino acids to choose to build the protein chain. This time, though, it extrudes the protein into the endoplasmic reticulum. The inside of the endoplasmic reticulum is very folded, so it has a lot of surface area. This is where the protein can be modified to suit its final purpose. The processing may continue into other organelles. Very often, proteins travel through the endoplasmic reticulum and then voyage through the cytosol to the Golgi complex. The proteins travel in capsules called vesicles, like small submarines. You can see the chambers and the different layers of the Golgi complex in this electron micrograph cross-section. A single type of protein may travel from one layer of the Golgi complex to the next, being further processed at each stage. It's here that carbohydrates such as glucose are attached to the proteins. Finally, its structure is completed and the protein can go and do its assigned job in the cell. The smooth endoplasmic reticulum has no ribosomes. Its function is to manufacture complex lipids, fatty substances. The mitochondria convert sugar to ATP or adenosine triphosphate. This is a chemical that is universally accepted in all cells as the most useful energy store. Again, the inside is folded to produce the maximum area for the reaction to take place on. Lysosomes float around in the cytosol, and if they come upon anything that should not be there, they engulf it like an amoeba. The lysosomes are essentially bags filled with acidic enzymes which are able to digest large molecules. Lysosomes also recycle waste materials. A typical cell often has very fine protein filaments running through it. These form what's called the cytoskeleton, which helps the cell to keep its shape. In many cases, cytoskeleton filaments contract like muscles so that the cell can move. The filaments are usually difficult to see with a light microscope, but they're quite clear here. They also provide a guide track for some of the molecules moving around the cell. Plant cells are the same as our model cell except for two important additions. Outside the cell membrane is a wall made of cellulose. Plant cells are therefore usually more rigid than animal cells. The most significant characteristic of plant cells is that they contain organelles called chloroplasts. The chloroplasts contain chlorophyll, which uses the energy from sunlight. It converts carbon dioxide and water into carbohydrates. You can see the green chloroplasts moving around this cell. Organelles that we haven't mentioned are the vacuoles. In a plant, they may take up most of the space in the cell. They are usually filled with fluid and form an important part of the structure of the plant cell. The typical cells we've been looking at have a nucleus with a membrane. They're known as eukaryote cells. However, there are more primitive cells which have no nucleus, but just a loop of DNA floating in the cytosol. They're called prokaryote cells and include organisms like bacteria. Cells which live on their own are called unicellular organisms. They have all the internal structures that we've been looking at. They can often move around under their own steam. They feed and control their water balance. Single-celled organisms are microscopic. We don't find a single cell such as a paramecium the size of a fish. A paramecium blown up to half a meter long would not be strong enough to hold together. And there are other problems. A microscopic cell is small enough for oxygen to diffuse quickly into the middle of the cell. But with bigger cells, it's more difficult. There is much less surface area in relation to the volume. Enough oxygen cannot reach the interior. As well, there is the control of the cell. This is done by chemical messages which travel through the cytoplasm. If the cell is too big, the messages cannot travel across the cell quickly enough, so individual cells tend to stay small. The question then becomes, how do organisms grow bigger? They do so by living in groups of cells, cooperating to form large multicellular organisms. Most of the living things that we are familiar with, including ourselves and our pets, are multicellular. In multicellular organisms, cells can specialize into different types of tissue which do different jobs. The living things on the earth are made of countless numbers of cells. Each one an amazing world of its own. For those of you who want to explore this further, here is a more detailed description of protein synthesis. The instructions for making a chain of protein are held in code on a particular stretch of DNA, and the operation starts when the DNA uncoils at this exact spot. The strands separate. Only one strand is used. Let's look at that again more closely. The DNA uncoils and its two strands separate. The instructions for making the protein are in code on the DNA. The letters for the code are the different chemical groups called bases on the DNA. We won't go into the details of the code here, but we'll see how it's used. There's not enough room in the nucleus to make proteins, so the instructions carried by the code on the DNA need to be sent outside the nucleus. It's done by taking an imprint of the code on a stretch of RNA. And how is this achieved? Well, free RNA bases are floating around in the nuclear fluid. They settle on the complementary bases of the exposed DNA strand. And they're then linked together to form a strand of RNA, which is like an imprint of the DNA. The strand of RNA now peels away from the DNA. It's called messenger RNA because it will carry a message of the instructions in the DNA. The messenger RNA carries the instructions out of the nucleus through one of the pores in the nuclear membrane into the surrounding cytosol. The DNA strands left behind can now coil up until the next time they need to make messenger RNA. The messenger RNA is now floating in the cytosol outside the nucleus. Also floating in the cytosol are the component parts of another set of organelles, the ribosomes. So we'll now find out what the ribosomes do. The ribosomes float around in two parts. That is, until they come across messenger RNA. The two parts then clamp themselves around the RNA to form a complete ribosome. Contained in the cytosol is a kind of soup of amino acids, some 20 different kinds. They're the building blocks of proteins. As the ribosome moves along the messenger RNA, it selects different amino acids from that soup and joins them into a chain of protein. The code of bases on the messenger RNA tells the ribosome which amino acid to select at each stage. And so, as the ribosome moves along, a chain of amino acids, in other words a strand of protein, is extruded into the cytosol. Let's now crack the code. The bases along the messenger RNA are read like a string of three-letter words. Each three-letter word tells the ribosome to select one particular amino acid. It adds that amino acid to the protein chain it's building and then moves along to read the next word, and so on. Let's look more closely at how the code is read. Amino acids are linking together and leaving the ribosomes as a chain, a protein chain, but they're not coming into the ribosome by themselves. So, what's happening? We'll look at our protein chain for a moment. Here we've got 7 different amino acids linked together. Here's one of the amino acids. However, it can't read the messenger RNA by itself. What it does is to latch onto its own reading device, its own transfer RNA. The transfer RNA has a code on it which matches up with its complementary code on the messenger RNA. It's the same kind of matching of bases that happens in the nucleus. In our model, the different colors represent the different bases. Blue will only match with yellow, and red will only match with purple. Each amino acid has its own transfer RNA with its own code of 3 bases, which match the complementary code of the messenger RNA. The transfer RNAs with their amino acids go into the ribosome and lock onto the correct spots of the messenger RNA. Bonds form between the amino acids and the transfer RNAs. Their job done, are released into the cytosol, and so the process continues. As the ribosome moves down the messenger RNA, a chain of amino acids, a protein strand, is extruded into the cytosol. The transfer RNAs are recycled. They go off and join up with free amino acids to carry on the process. The production cycle is going on all the time, with protein chains being made by free-floating ribosomes. The protein chains are released directly into the cytosol.